Thank you for coming, especially for students. Today is Sunday. They have to come from long distance. One of the students I just learned come from Nanjing. Is have to take 2.5 hour flight to come here. And the student, that moved me. I did exactly the same when I went to see Alexander Rich. Took 6 hours flight from Santa Barbara, California to New York to meet my mentor, Alexander Rich. So the take initiative for students is extremely important for seeking new careers and to do great things. And several people have told me brief life stories. I just take a few minutes to tell you mine. I did not go to high school, just 6 years elementary school, then went to college. I was a terrible student in college the first year, finally barely got through. What got me through is I asked a lot of questions. Because when I was before the college, I was in the military. Ask questions in the military was forbidden. I got punished and a terrible soldier, demoted. First feeding horses, then feeding pigs, finally become a cook. Because they thought they didn't want me to ask any question. But in science, everything still is ask questions. So here is the first slide. And Schrödinger said something. The task is not so much to see what no one has yet seen, but to think that what nobody has yet thought about that which everybody sees every day. So I will tell you something and today about my research. Follow this path. But before that, I will show you, here is MIT Media Lab where I work. IMP designed the building in early 80s. And he designed MIT into the building. How many people here in the audience from MIT never noticed that? This is M, this is I, and this is T. Many people walked hundreds of times, 1,000 times, didn't notice that until you pointed out. Later, he was recruited to design the Louvre of the Paris. Here is Alexander Rich, my mentor. is Francis Crick, Watson, and Leslie Ogle. The photo taken 55 in the house. The golden helix, many people in Cambridge have seen that, was installed 1951 when Linus Pauling just published the structure, gas structure of alpha helix. 2 years later, Francis Crick and Watson, Rosalind Franklin, and Wilkins discovered structure double helix. Crick has every right to replace that as a double helix. He didn't. This photo was taken 2023, April, when I went to Alan's birthday party. So today's talk has a lot to do with this alpha helix. Crick came to visit Alex in 1988, but he always tell people, you should always ask questions. The bigger, the better. If you ask questions, you get big answers. AI cannot ask questions. LLM, ChatGPT, cannot ask questions. Google cannot ask questions. Wikipedia cannot. Only you has a brain, real brain, can ask questions. That's how science life starts. Here is Bob Langer. He said in school, you are judged by the question you answer. In life, in real life, you are judged by the question you ask. That's absolutely true. When I interview student postdocs, not only I ask them questions, I ask them to ask me questions, you can tell very quickly the people is right person or not. Here's the questions. Yeah? Should always ask questions. Show you, stuck. So the question is, what do they have in common for high school student? For the student here, what do they have in common here? The clouds, the Amazon River, the tree branch in the winter, and the drought, and the waterfall, broccoli, and the human circulation system. What do they have in common? Anybody? Yes? Perfect. Yes, fractal. Very good. The father of fractal, Benoit Mandelbrot. He defined fractal as self-similarity at multiple scales with simple equation here written. It can explain so many complex systems no other mathematics, no other physical system can explain. Fractal is also art. And here, this one here on the right, it looks just a regular fractal. However, it's very crucial for your mobile phone. We'll show you that. Here is a scale. The same scale is measured by billions of light years. The same bar, look at the bar. The next one is a nerve cell, neuron system. The same bar, the 100 microns. This bar, 1 micron. The same scale, bar, but a different scale. This is my own research discovery, self-assembling peptides. You'll hear more this afternoon by Professor Zhongli Luo. Here is, look at your phone. For people who were older in 1970s, 1990s, every phone has an antenna. No more antenna. Where did antenna go? Inside your phone with a fractal receiver for the signals. That's the fractal. Here is my talk. The QTY code for protein design. I always ask questions. Started from the question from Alex Rich. He asked me in 2010, can you convert a hydrophobic alpha helix into a hydrophilic one? In December 2010. And my answer was to him, yes, immediately, because nature has done it. Hemoglobin and myoglobin are water-soluble alpha helix proteins, but membrane proteins, most transmembrane proteins, alpha helices, photosystems, GPCRs, transporters, they're not water-soluble, but they are also alpha-helices. So the question set me to do something unusual. And in those days, early days, people consider I'm crazy to do that. So those are, before I forget, I want to give credit to my student and postdocs that did early work on QTY code. So Carolina Coren, Loda Tegler, Tang Hongzhi, Fei Tao, and Ray, he's in the audience here. And Michael Skursky. By the way, Joe Ito said 60% MIT student has spectrum on the ultimate spectrum. He's one of them, but a brilliant student, High House Lay. Here are two classes proteins. One is water-soluble. The other one is water-insoluble. Water-soluble one, hemoglobin, you have seen the photo several times, is fully water-soluble in the blood. The alpha-helix protein, membrane protein here, they have exact molecular structure, but they're not water-soluble. But the question is how to convert them from water-insoluble one, membrane proteins, into water-soluble form. That would consider a crazy, impossible idea. Carl Brandin, one of the colleagues, wrote a textbook. First edition 1991, second edition 1999. I use for teaching, use the textbooks. So he classified the alpha-helices into 3 kinds. And he didn't explicitly say, but this alpha-helices is the most hydrophobic, the green. This is half and half the green here, the other is water-soluble amino acid, there's water-insoluble amino acid. The third one is most of water-soluble form. So in 2022, Bank asked me to write an article about my idea, a QTY in the QRB. So I wrote, classify specifically alpha-helices in 3 kinds. We call it type 1, water-soluble kind, type 2, water-insoluble kind, membrane kind protein. Third one is type 3. So Linus Pauling described alpha-helix in 1951, 1920, 22, 7 years later. People have seen alpha-helices for 70 years, did not specifically classify them into such form. And I was struck by this image from David Eisenberg's teaching website. David Eisenberg is an excellent X-ray crystallographer. He used it for teaching undergraduate students how to each amino acid size and shape. So here is first time I saw the textbook, not textbook, on the web, show arrange amino acids by the size, not by the chemical properties. So once arranged by size, something immediately jump out. It's here, those two. This is Valine, this is Threonine. Water-insoluble, water-soluble. Second is Leucine with aspartic acid, asparagine, glutamate, and glutamine. Those two are charged, this one is not. Finally, is Phenylalanine with histidine and Tyrosine. This is amazing image, but people didn't notice that. That's also amazing. So we know in textbooks the 11 amino acids, the water-soluble, others are not water-soluble. So in water-soluble kind, they can form 4 hydrogen bonds, thanks to Phil Ball's book, Life's Matrix. By the way, I also use that for teaching. And form 3 hydrogen bonds because each water molecule can form 4 hydrogen bonds as donors, 2 donors, and 2 acceptors. So those amino acids can form 4 water molecules surrounded side chains. This can serine, threonine, and tyrosine OH group can form 3 water molecule hydrogen bonds. There's arginine 5, and there's lysine, and 3, and histidine 2, and tryptophan 1. Those two form, this one has charge, but this one has no charge. So in order to not alter protein surface, I selected the Q here, Q, and the T, and the Y. For Y is here too. So I call the QTY code for changing protein systematically. So idea is to change membrane protein embedded in the membrane. If you take them out, you need detergent to surround it. Otherwise, protein collapse in a few minutes. But the idea is transform membrane protein into water-soluble form. It's considered impossibility. And because for 70 years, not many people working on this, except one person, Bill DeGrado, is a pioneer in this area. He started to alter membrane protein into water-soluble form in 2003, published paper. But this is the best one here. He changed amino acids, this native protein, crystallized into this structure, is peptide, a 25 amino acid peptide, into this structure. And he altered this, changed several of them from leucine to glutamate, and isoleucine into lysine, arginine, and so on, and leucine into glutamate, so on. But Bill did the change through the computer calculations. And this is not a change, is not a one-to-one change, because leucine can change to several different amino acids. And it's not made, it's daunting project to do this non-systematic way. But Bill is pioneer, did this work, you can see. So I diddle-daddle on a piece of paper, and then how to change from hydrophobic into hydrophilic on January 15th, 2011, after Alex Rich asked me the question 3 weeks later to write the NIH grant applications. Finally, I listed V2T, I2T, L2K, QI, F2Y. This is the aha movement that came out, QTY, because it was specifically no charge for the proteins. That's what we did, apply the QTY code with membrane proteins, and this is QTY code. Leucine changed to asparagine, or Q is glutamine, and Isoleucine, Valine changed to Threonine, and Phenylalanine changed to Tyrosine. In the transmembrane domain, this membrane protein called CXCR4, and with not hydrophobic residues, the green color. With applied the QTY code, we change into water color, cyan color of water-soluble amino acid, QTY. Very simple, one-to-one change. So here's a cartoon, here's an animation, shows this membrane protein inside, green, is a dimer. We make it now change into QTY, now beginning to change into water-soluble color, become water-soluble. Now the protein can no longer stay in membrane, they now stop. If it came out of membrane. However, they can still bind the ligand. That's experimentally proved. The same ligand binding is very similar as a native protein. That paper was published in 2018. It's like a hand. You wear gloves, you have become hydrophobic, the glove, or this is hydrophilic. But structure is the same, the function is the same. Surgeons, they have to wear the glove to operate in people. And this one, you know, you don't have to wear glove, can carry out the same function. So this will be the membrane form, this one by QTY form. The ligand binding is called the movement of truth. And the binding ligand for the protein QTY version, the ligand binding is very, very similar, not only in water, but also in human serum. The binding, the affinity is very similar. Here is another one, so CXCR5, CXCR4, you can see binding in water, binding in human serum. Okay, so Michael Skursky, the student here. By the way, he didn't go to junior high school, didn't go to elementary school, didn't go to high school, didn't go to university, didn't go anywhere. His only degree for his entire career is MIT degree. Master MIT and PhD MIT without the undergrad degree. So he's like Zhao Bowen. He did not drop out, he just simply did not go to university, but got a 2 degree from MIT. So he's a computer genius. He simulated, and before AlphaFold2 came out, simulated each protein take 6 weeks each. But when AlphaFold2 came out in July 2021, each one take 2 hours to simulate, to predict by AlphaFold3. You have heard AlphaFold3 yesterday. Now AlphaFold3 came out, even more powerful. Instead of 2 hours, now just 3 minutes. So you can see one with the green color is a native GPCR. And then the cyan color is the water-soluble QTY version. it's amazing similarity predicted by AlphaFold2. This is another GPCR adenosine receptor. You can see it's a native structure. Here is not a yellow color, such as hydrophobic surfaces. If you take those native protein out from the membrane, you must have detergent surrounding them. Otherwise, they will die very fast. The QTY proteins, we have purified them now more than a dozen. My Qinrui has purified another dozen, so more than 2,000 protein purified without any detergent. All the experiments carried out without any detergent. People say, what's the big deal without detergent? It is a very big deal. Now the protein, once you purify into water-soluble form, those proteins soon can be useful for biotechnology as a decoy receptors or as a sensing device. So we not only did the GPCRs, but also did some olfactory receptors and others use in the pink color, magenta color, is cryo-EM structure of olfactory receptor, the 3 of those. And the green is the native one predicted by AlphaFold3, and the cyan is AlphaFold3 predicted by AlphaFold3. No, the 3 is the QTY version. And apply the same tool, not only now we don't need this cryo-EM structure. We can take the native protein structure and QTY to say the structure remain very similar. Not only that, the predicted, and the people have done experiment showed the ligand binding. The ligand is an odorant molecule. You can see it's a hydrogen bond pattern, and a negative charge odorant bind to positive charge residues in the receptor in this area here. And the hydrophobic areas from the hydrophobic residue chain bind to the odorant. Likewise, another one called the polyamine receptor is also olfactory receptor type. And in this case, the molecule is polyamine, is positive charge, is amine. Now the negative charge and the residues in the receptor here, then bind to the positive and bind to amine area, this spermidine. And the hydrophobic residue bind this area. We did it not only by AlphaFold3 prediction, but also the molecular simulation, molecular dynamic simulation, because odorant bind to odor, the smell so far still has not been fully understood at the chemical level. The student here, if you want to pursue science, lots of people asked the question yesterday, to find something that's doable, but take a longer time, take persistent, what is good to work is understanding our smell. We have done our editing the genome, we can go to the moon, we can go to the deep sea, but something we don't understand how we smell. This is a doable project in next 30 years or 20 years. So young people should think about long-term project to do that. So the QTY code not only applied to the GPCR, but also to other membrane proteins. This one has a 14 transmembrane domain. And if you see how much you have changed, is total 26% of the total protein. But in transmembrane domain, is significant, it's here it says 48% change in the transmembrane domain. But the PI change is minimal, and likewise is this one here. So change 44% transmembrane domain. Only on the blue part, change from L to Q, and I to T, V to T, and so on. And look, the molecular weight change is minimal, and pI change 0.02 points pH, because we deliberately use glutamine, tyrosine, and threonine. So this is a glucose transporter, and the magenta color is cryo-EM structure, and in front, back, top, and bottom, and those two are the same, two different glucose transporter, 1 and 3. You can see the close proximity is amazingly close. Here it's a student did this. I'll tell you later about her pursuit. So there's a native structure and need a detergent to keep them alive. There's a QTY version for that. And no detergent needed. And one of the students, high school student, okay, he also did work on this called ATP transporters. ATP transport is essential, one of the oldest transporters in cells. And the magenta color is cryo-EM structure, and the cyan color is QTY. But you can see the structure is remarkably similar. Sorry. Battery come out. But you can see it's a native structure with a lot of yellow part, and the other one is QTY version, yellow part significantly reduced. Oops. This is a Turkish brothers in Istanbul, medical student. They wrote me a letter out of the blue in 2002. They asked me, can I take your class online because of COVID? I said, yes. After taking my class, they asked me, can I do research with you? I said, yes, why not? Because the internet allows students to do some remote research. So then medical doctors type, they want to study serotonins, dopamine transporters, and so on. So that then did the work. Again, in magenta is a native structure, and cyan is a QTY version. On the left, it's a native structure, and on the right is QTY version. You can see all this alpha-helices works well. That's why Francis Crick, that house alpha-helices, is directly applied to our research. So we asked, would the QTY also work for the enzyme? So far, we work on receptor transporters. If we work on enzyme, would the enzyme still carry its enzymatic function? If enzyme is dead, this whole QTY code is useless. And interestingly, the enzyme, by the way, biodegradator also work on this enzyme, bacterial histidine kinase. It's a dimer in the native form, and this is how we change native form into QTY version. You can see that's how much has changed, and a lot amino acids changed, 17 something, 1 to 12. What's most interesting is here. In the native structure, it's two hydrophobic residues interacting. And 17, A is dimer, numbers A is L17 and B17 hydrophobic interactions. And valine and leucine interactions on this position, 24, 21. And leucine and isoleucine, 182 and 112. Now, if you look at the QTY version protein, at the exact same protein, instead of hydrophobic interaction, now they have hydrogen bond interactions. Here is 2Q interact because leucine changed to Q. Here is the T and Q interactions. It's valine and leucine changed interactions by hydrogen bond. And finally, is leucine changed to Q, and this is changed to T. You can see hydrogen bond interaction at exactly same locations. The paper was published this year.
actually should have already been published. So enzyme remain fully functional, has the 4 enzymatic functions into protein kinase activities, phosphorylation activities, transphosphorylation activities, and cell cycling activities. So every single one was preserved. The next question is, as I told your student, always ask questions. Because protein has 2 motifs, major motifs. One is alpha helix, which we described. Second is beta sheets, the lot beta sheets proteins. One of them is antibody, Professor Greta Winter has mentioned about. And other is bacteria transporter. So this is one of them. You can see this taken from PDB. And the green is crystal structure, the protein. The cyan is the QTY version. It's amazingly close, the structure. And this is native one. This is the QTY version. So why study those proteins? Because bacteria poring and bacteria, the beta heats frequently are involved the drugs resistant. Bacteria drugs resistant, bigger problem in the hospitals. So you can use this to study antibiotics, discover new antibiotics. Somebody also mentioned it yesterday. Leonardo da Vinci's the simplicity is ultimate sophistication. So several people mentioned DNA double helix. A few people even mentioned it's a code. It is a code, DNA. Once you know one side of TCAG on one side, other side is automatically known. It's complementary. So the simplest is DNA is a code. So now I believe DNA is a code. Also, like QTY code is also a code. The change is 1 to 1, L to Q, Q to L, reversible, just like DNA is reversible. And valine isoleucine to T, phenylalanine to Y is reversible. So this paper was published in QRB 2 years ago. So next question people ask, so what? You have the membrane protein got water soluble, would be useful. So in collaboration with Uwe Schleiter in Austria, in Vienna, he discovered called cellular proteins here. Cellular protein is a bacterial surface protein. It's actually, if you look closely, is 2-dimensional crystal on the bacterial surface. So the distance between those dots here, white part, is 13 nanometer, very organized, arranged on the surface. So in collaboration with Professor Uwe Schleiter, by the way, he discovered this in 1968, totally curiosity-driven research. And by accident, he discovered, pursued for 50 years this research, 50 years on this. Here it's, if you usually lay down molecule on the surface, the molecule on the surface totally rundown, has no orientation. But now cellular protein is a yellow one here, and now you lay down molecule because it can be linked to the molecule. Now everyone is 100% upward. So that is very important for engineering. So this is the idea here. This is experiment to show you how to do it. So the yellow is cellular protein. Red one is our receptor. The ball is the ligand binding. Now you can make a device, the 100% upward, which is extremely useful. Qin Rui, his audience, and other student, with collaboration with MIT, physicist, electrical engineering, made a device. The device then called the scalable biomimetic sensing system with membrane receptor, dual monolayer, and graphene transistor arrays. We call them called RESENSA, and it's the acronym. So how we did this, is Ray used to change it to QTY version of 6L4 and covalent link to make it into an antibody-like structure, remove FAB, but linking this receptor to FC domain. Now look like antibody, but not quite an antibody, but the receptor now can recognize the ligand. Now you couple with estriol protein on two-dimensional surface. Each 13 square nanometer can accommodate 8 receptor 100% upward. That's very useful. So now make a sensor here, 16 by 16, and now it's 256. That's individual sensors. You can now do the measurement ligand binding. As low with regular ligand, kind of measure just like other machine, SPR or micro scale thermal system, thermophoresis. You can see can really take a measurement of ligand. Not only is it's only proteins, 6L4 ligand, but also HIV infection enters cells through the 6L4, can measure down to 10 nanometer for the protein. And because this electrical signal with this as a tip here, its computer reaction is much more sensitive than SPR, much more sensitive than an MS microscale thermophoresis system and other system. What's important is all the signals are clustered. It can easily be analyzed by a computer. Insulin is not the receptor for the 6L4, so it doesn't bind at all. This is the latest research. Ray and other Housley did experiment called molecular trap. Molecular trap, somebody had mentioned the decoy therapy. It makes something, membrane protein usually stuck in the membrane, then cannot come out. But once you make a water-soluble version, then come out, then can soak up all the molecules at 6XCL12, and now not only can do that, but also now reduce 6L4 cycloning and reduce 6L4 exposure to the cell surface. Now the increased cell apoptosis and the reduced metastases, cancer progression, immune rejection, and drug resistance. If have not made the 6L4 in the water-soluble form, we call the QTY, FC domain coupled, it's impossible to do such experiment or such measurement. So this is data for animal then did in Chongqing and in Shanghai. And then both spend time in my laboratory. And forget about all the mouse, it's complicated to see. Just see that before the treatment, this FC, QTY FC treatment significantly reduce the metastases. You can just say the animal after several weeks have almost no or little the metastases occur in the system, in the for cancer in animals. So this is Ray, and draw a cover of this chemical review article by this student. I will tell you more later when the question answer. The student also, it's amazing, wrote me out the blue again. Here is my favorite quote. Francis Crick said, "One could not be dedicated to anything unless one believes in it passionately." And when I started QTY research, most people don't believe it. And luckily, the students either not have enough experience, undergraduate student, or then also passionate about the science, then found my foolish or foolish, like Steve Jobs said, always stay foolish, and doing pioneer research that require passion. Okay. What I also want to say, have a few minutes, 20 seconds left, is Francis Crick, the double helix on his house, not double helix, alpha helix on his house. The protein alpha-helices has been known in the water-soluble version and in the membrane protein. Not many people had paid detailed attention to this. And it took me, or took other communities, many, many years, decades, to now can now freely convert the hydrophobic membrane proteins into hydrophilic membrane proteins. Thank you very much.